So good morning, grade 12 students. In this video, we're going to start solving the genetics exercises for probing the documents and exercises for the chapter. So here we'll start with probing the documents on page 61. What I want you to pay attention to this. Here we have the non-transcribed DNA strand of a normal gene that is found here and its corresponding amino acids. Here we have the non-transcribed one. So, and then we have the mutated one, the non-transcribed strand of the mutated gene and the corresponding amino acid. So there's the non-transcribed. So Ali, non-transcribed is called what?
Non-template
and it's
coding.
the codon. So I found the sequence of the, let's say, of the m-RNA. What should I do if we have the non-transcribed?
It's the same, but replace T by U.
Excellent. Replace the T by U. While if we have Muhammad, if we have the transcribed, what do we call the transcribed?
It's called the template non-coding.
Non-coding. So if you have found the sequence of the mRNA, I should make it complementary, but instead of T, it should be replaced by U. Excellent. So Hassan Watfa. Yeah, let's start first with the first sequence. We have the non-transcribed of the normal strand of gene and the corresponding amino acid. So we have CCA, then we have CAG, and we have ACT. So this will go the amino acids pro, glu, and THR. These are the sequence of the normal one. Okay? So these are given according to the genetic code table. Now we move, Hassan, for the mutated one. The mutated one, as you see here, it's substitution. So substitution, they are of three types. Either they are missense, or silent, or nonsense. So let's start with the first one. What is the position of the mutation in this sequence? Show position and mutation, ya Hassan. So we have here the first codon, the second codon, and the third codon. So what is the position of the mutation? Which codon? So this is the first codon.
The GTA second.
Second one?
Yes.
The second codon. So you have to specify the second nucleotide of the second
Codon.
Codon. Excellent. So here we have the same CCA, here we have CCA, the same. GAG-A is substituted by T. So this is the type of mutation. The mutation is found in the second nucleotide of the second codon. This leads to the change in the amino from glu to valine. So VAL. So here we have the change in the sequence of the amino acid. So this is called missense. Right? Mutation by substitution. Let's move to the second one. Second sequence. Here we have CCA, CAG, and ACT. This is the sequence give pro, give glu, and give THR. So here, Dana, what is the type of mutation here between these two? What is the type of mutation? Sure, it's substitution, but I want from you to indicate the position of the mutation. What is the position of the mutation? In which nucleotide and in which codon?
And the sixth one, we substitute A by...
Okay, so I want you to specify the nucleotide and the codon. First of all, which codon?
The second one.
Second codon. Which nucleotide of the codon? The third nucleotide. So the position is the third nucleotide in the second codon, where it's a G in the normal, and it's substituted by A in the mutated. So here, did you see here the sequence of the amino acids pro? It's a pro. Glu, it's a glu. THR, it's THR. So the sequence of the amino acid is not changed. So there is no detectable change. So what do we call this type of mutation? It's called point mutation by substitution, but it's silent mutation. Okay, thank you, Dana. Let's move now to Hadil. Hadil, the third sequence. The third sequence, we have the normal, CCA, CAG, GAG, and ACT. In the abnormal, we have a mutation at this position. Can you indicate the position of this?
First nucleotide of a second codon.
Excellent. So this will lead to the stop codon. Whenever it leads to the stop codon, what do we call it? It's called a nonsense point mutation. So the stop codon will stop the amino acid sequence. So it will lead to the smallest amino acid sequence, and it's called the nonsense point mutation. So let's move now to, let's go back to Rama. I think she can speak. Rama, good morning.
Good morning.
Good morning. So this sequence, we move to another sequence or another type of mutation here. It's deletion. What's the meaning of deletion, Rama?
Delete something.
Delete one of the nucleotide. So here we have the sequence, the normal one. We have TAC, ACC, ACG, A. While here we have TAC, and what happened here? We deleted the A here. The A is deleted. So the sequence will be shifted to the left. So it will be CCA, and then we have CGA. So here the type of mutation is frame shifting mutation by what?
Deletion.
By deletion. Okay. For the third, the last one. The last one is the type of mutation is insertion. What's the meaning of insertion, Yarama?
Insert one
Insertion of one of the nucleotides. So this insertion occurs between the third and the fourth nucleotide, as you see. So the first nucleotide, TAC, it remains TAC in the mutated. But here we have insertion of G here. So it will be GAC, GAC. Then we have CAC, CAC, and then we have GA. So this is called frame shift. This is shifted to the right or to the left. Okay? So this is the types of mutation for this part and following the document. So we solve parts 1, 2, and 3. So part 1, what did they ask for? They asked for the first sequence. So we said that the non-transcribed is called the non-template. It's the coding. So if we want to find the transcribed, we should make it complementary. m-RNA, it's the same as the non-transcribed, but instead of T is replaced by U. Why? If it's transcribed, it should be complementary, where T is replaced by U. Okay? For the mutated one, as you see here, mutated, so the amino acid sequence changed from glue to valine. So the amino acid we have changed in the amino acid sequence. So this is called missense point mutation by substitution. For the second one, or explanation for the first, a change in the second nucleotide of the second codon. So here we have to specify the position of the mutation. Second nucleotide of the second codon. And you have to pay attention in the exam if we have number of nucleotides or we have codons. Okay? So A is substituted by T. So this change in the amino acid sequence from glue is substituted for val or valine. Second one, the silent mutation. So mutation it took place in the third nucleotide of the second codon, where it was substituted for A. This leads to no change in the amino acid sequence. So they form the same amino acid sequence. So the formation of protein, it will not be altered or changed. The third one, we have deletion. So we deleted one of the nucleotide. So we have frame shift. So it's going to be shifted. Following the document on page 66. Pay attention here. We have this DNA molecule. So we have cleavage of the DNA molecule. We cut this DNA molecules by two different restriction enzymes. This is the first restriction enzyme, and this is the second echo R1 enzyme. So first of all, what I want from you is to pay attention to this. First, when we did restriction enzyme, let's give the symbol for the restriction enzyme RE, restriction enzyme. Restriction enzyme, it cuts the DNA into how many parts? Let's go back to Ali. Ali, how many parts of the DNA here we have?
4 parts.
4 parts. So we have 4 fragments. This is 1, this is 2, this is 3, and this is 4. Do you remember the rule? Number of fragments or do you remember the rule for the restriction enzyme?
No.
Who can give me the rule for the restriction enzyme and the number of fragments? Yes? This is the rule. The rule is number of fragments. The original rule, the number of fragments is number of restriction sites plus 1. So here we have the number of fragments. So number of restriction sites is equal to the number of fragments minus 1. For example, here we have how many pieces? We have 1, 2, 3, 4. How many times did I cut? If you have a paper, I want to cut it into 4 pieces. How many times should I cut? Yes, Ali.
3 times.
3 times. So the number of restriction sites is 3.
3.
While the second one, let's move now to Hassan. Hassan, what for? The restriction enzyme echo R1. We cut this DNA by the restriction enzyme echo R1. How many fragments did we get?
7.
1. Let's count them. Yeah, you said 7. 1, 2, 3,
Yes.
4, 5, 6, and 7. So we have 7 fragments. So number of restriction sites is how much? It's always -1. So how many restriction sites? We have 7 fragments. Number of restriction
6.
sites, the position where we cut, it's equal to 6. That's it. So recognition site of echo R1 is repeated 6 times. Why? Because this enzyme cuts the DNA into 7 fragments. Why? Not 1 is repeated 3 times. Why? Since we have 4 fragments. So the original rule is going to be like this. This is the original root. So original root for this is like this. It's going to be number of fragments. It's equal to what? It's math here. Number of fragments.
Number of restriction sites
Plus
over 1.
Plus 1. So
plus
number
1.
of restriction sites, let's do it restriction sites or as plus 1. So if I have, for example, 7 fragments, what is the number of restriction sites? Going to be what? Math is going to be 6 because we have to add it to 1, so it's going to be 6. This is a simple example. This rule, you have to study it. Number of fragments is equal to the number of restriction sites plus 1. If I have the number of fragments, I can calculate the number of restriction sites, and the opposite is true. Okay, guys?
Okay.
All right, so let's move now to document C. Document C is showing the electrophoresis of 2 different alleles. They are cleaved by the same restriction enzyme. So here we have explained the results obtained on document C and what causes this difference. We have the restriction enzyme. How many times the restriction enzyme is repeated here? Let's see, 1, 2, and 3. So we have 3 times the restriction enzyme is repeated. While here we have 2 times the restriction enzyme is repeated. Why? Pay attention here. The sequence is GGA-TCC. This is the sequence where the enzyme is going to cleave, it's going to cut. And here we have the same sequence, GGA-TCC. And also here we have the same sequence, GGA-TCC. The second one in the part C, or in the fragment C, we have GGA-TCC. And here we don't have a GGA-TCC, we have AGA-TCC. So the enzyme, it will not cleave here. So we missed one of the cleavage sites. And the last one, GGA-TCC. So here we have mutation. So what do we call this type of mutation, Yadana? Here we have GGA, it's on AGA. So the G is substituted by, yes, Dana? The G here is substituted by A. So what do we call this type of mutation? Okay, Dana, you can speak. Now you can speak, Dana, I think.
Yes.
Yes, Dana. So what do you think? The G is substituted by A. What do you think this type of mutation is?
A frame shift mutation insertion.
Wait a minute. G is substituted by A. So G is substituted. So what is the type of mutation? Substituted. That's it, substitution. It's not deleted or neither inserted. Okay? So here what happened? Allele 1 and allele 2. So allele 1, this is allele 1. This is the 1. How many bands do we have here, Ya Ali? Allele 1. Ali Hariri, how many bands? Here, allele 1. Pay attention.
2 bands.
2 bands. Why allele 2?
Has 1 band.
Has 1 band. So this is for allele 1. It shows 2 bands. So here, cleavage moving from the negative to the positive. Pay attention here. The one that stays beside the negative should be the heaviest one. And here, the heaviest one, it can't move. So the heaviest one is here. Which one? A, B, or C? Which band?
C.
C is the heaviest. Why? Because it stays beside the negative. So it doesn't move towards the positive pole. So it stays beside the negative. يعني هي أتقل واحدة ما بتقدر تمشي لتحت ما قدرت تمشي. So this is the heaviest one. Then after C, what do you have? Which one the heaviest after the C, Hassan?
A.
A. And which one is the least one? Yes, Hadid, what do you think? We have C is the heaviest. Then we have A. And the lightest one is going to be the B, since it moves toward the positive pole. So here, gel electrophoresis, it shows the bands here. And pay attention as a note here. Whenever we have thick band, meaning that you have two, they are similar bands. So for example, if you have thick band in C, for example, thick band, so meaning that we have two for the C. So if the thick band indicates the homozygosity. We'll talk about this. So allele 1, it shows two bands after being cut with the restriction enzyme BAM H1. Since the recognition site is repeated how many times? It's repeated 3 times. So we have two bands. While allele 2, we have a mutation that we call it Yadana substitution. So here goes the appearance of the restriction site. For example, for 1, 2 times. So we have one band is found here. So we have one fragment only. So there we don't have cutting at this region since we have a mutation by substitution. So in question number 3, referring to the document D, this document showing the electrophoresis of DNA fragments, they classify the fragments of document C according to their decreasing length. So here, decreasing length, we have 100 base pair. This is for the length. And this we have 200 base pairs. And we have here 300 base pairs. Moving from the negative to the positive is going to be the heaviest one. There's one that stays beside the negative. So direction of movement is going to move from the negative toward the positive. So here, the heaviest one is the one that sits beside the negative. And the one is least heaviest or the least one is going to move toward the positive. يعني أتقل واحدة راح تضلها فوق. والخفيف is going to move toward the another pole. Okay, so this is what's known by the size of the fragments. So band C is the one that's closer to the negative pole that we answered already, than band A, than band C. So band B is this one, the most far one from the negative pole. So the DNA represented by band C migrates less than that represented by band A and less than that represented by band B. So band C is the larger one, then band A is the larger, and then the least one is going to be B. So the last one, the most one, 300 base pair is going to be for C, 200 base pair is going to be for the A, and the least one, B, is going to be 100 base pair. This is the note that the thick band, it represents 2 copies for the allele. It's said to be homozygous. So let's move now to exercises on page 72 on your book. Exercise 2. Let's move to Rama. Rama, you have to use the genetic code table to answer the following. What is the amino acid sequence of the peptide translated from the following DNA strand? Here, DNA strand, they didn't mention whether it's transcribed or template. I'm going to use it as template. So template, Rama, is what? What do we call the template?
Transcripted non-coding.
Transcribed, and it's the non-coding. So if I found the sequence of the m-RNA, what should I do? I have to make it
Coding.
I have to make it complementary, or I have to find the coding, then I can find the sequence of the m-RNA. But to save time, if you have the template, you have it's the transcribed, it's the non-coding, so I can make it complementary, and instead of T is going to be replaced by U. So here I have the template. I have to find the sequence of the mRNA, so it's going to be complementary. T, the complementary is A, while A, the complementary is T, but there is no T in the mRNA, it should be replaced by U. C, it's G, T, it's A. It's easier or not, Rama? Instead of giving the coding and make it complementary and then finding the sequence of the mRNA. It's easier and it saves time here, right?
Yes.
Okay, so you have 2 options. If I have the non-template, the non-transcribed, it's the coding, same sequence, instead of T is replaced by U. If I have the template, the transcribed, the non-coding, I have to make it complementary, while instead of T, I have to replace it by U. Here you save time and you can give the sequence of the amino acid easily. Okay, so here we gave the sequence of the amino acid. So we have met, ILE, ALA, cis, CER, and then we have the stop codon, and this is the amino acid sequence. Part B, what did he ask for in part B? How would this deletion of a nucleotide at position 5? So let's move to Hadin. Hadin, they said, how would the deletion of a nucleotide at position 5 affect the sequence of this polypeptide? So affect the sequence of the amino acid. Where is position 5? Can you give me what nucleotide is found at position 5?
A.
A, excellent. So here at position 5, it's A. So you have to pay attention here. They are mentioning the position 5 and they give you the number of the nucleotides. Mahada Bukr, you know that this is the 5th codon. How come 5th codon? This is the 1st codon, TAC. 2nd codon is TAA. The 3rd is CGG. 4th is ACA, TCA, and AGT. For example, if I ask you, Hadil, what is the nucleotide found at position number 13? Can you find it easily?
T.
T, excellent. So at position 10, it's A, 11, 12, and here 13, it's T. Excellent. So here they deleted one of the nucleotides. So they deleted the A. So the sequence becomes TAC, TAC, GGA, CAT, CAA, and then we have TT. I have to find the sequence of the m-RNA complementary since it's the template, but instead of T is replaced by U. So this mutation, what does it lead? It leads to the disappearance of the stop codon. So this will lead to the change in the amino acid sequence. And we have a stop codon. This stop codon, it's disappeared. So the amino acid sequence will become larger, and as long as the translation doesn't stop at this stop codon. Exercise 5. Moving to exercise 5, a mother thinks that her child was exchanged for another at birth in the hospital. So بتعتقد ان ابنها تبدل بالمستشفى اول ما خلفت الولد، بدلوا لها اياه بالمستشفى بالغلط. The document below represents a DNA fingerprint performed to find out the truth. First of all, we have the bands of the father, we have the band of the mother, and we have the bands of the child. Has her child been really exchanged for another? Justify your answer. So what do you think? You have the answer. Yeah.
No.
Hassan said no. Why?
Since the child had the 3 bands of his parents.
3 bands. He has a common band with his mother or not?
The
this
middle
band,
one, yeah.
Yes, this band is common between the child and the mother. So the child has been not changed. It's not changed. So he had a common band with his mother. So there is no change. So restriction map of the mother and the child, so a common band. So DNA of the child matches that of the mother. Should be the biological mother. هتكون أمه البيولوجية. So the child hasn't been exchanged. Exercise 6. The sequence TCT AAA TGG is one of the possible sequences of the DNA coding non-transcribed strand. هون هونا عليك قال لك DNA coding وقال لك non-transcribed. So بالامتحان يمكن قال لك أنا the coding. تيجي تقول لي أنت أي sequence says أي بدي استعمل. You have to read the question carefully. It's the coding non-transcribed. So if I have to find the sequence of m-RNA زيات, what I have to do?
This is the non-transcribed.
Non-transcribed.
It's similar.
Okay.
mRNA is similar to the non-transcribed, but T is replaced by U.
Okay, so it will be, can you give me the sequence? We have TCT AAA TGG. Can you give the sequence of the m-RNA?
Yes.
Yes.
UCU.
UCU.
AAA, UGG.
AAA and UGG. Very good. So what are the different types of gene mutation? What are you have different types of gene mutation? Yes, Hariri, what do you think? Different types of mutation.
By substitution.
Substitution, which is of 3 types. شو هن؟
Missense. Nonsense.
And?
Silent.
Okay, and then the other types we have deletion or?
Insertion.
Insertion. We call them frame shift. How can you obtain silent mutation in this case? Silent mutation, whenever we change one of the nucleotide and still it gives the same amino acid. Identify the possible mutations. Yes, Hassan. Muhammad Zayyad raising his hand. Yes, do you want to ask a question, Muhammad?
Yes, I have a question.
Yes.
إذا بالأمينو أسيد صار في deletion بالبوليبيبتايد.
Deletion, it occurs mainly in the sequence of the nucleotides and the nucleotide sequence.
إذا صار في deletion بشي nucleotide وهيدي أدت لستوب كودون.
Yes.
نحسبها nonsense أو نحسبها deletion.
Deletion here of almost is going to be deletion. Almost. Nonsense whenever it's substitution or we substitute one of the nucleotide. Whenever you have deletion, always is frame shift mutation by deletion. Always. Okay? So identify the possible point mutation which lead to a stop codon. Let's talk about these three kinds. We have deletion, insertion, and substitution. We said that substitution we have missense, nonsense, and silent. So here we have silent mutation whenever we change one of the nucleotide and causes the same amino acid sequence. Stop codon whenever we change one of the nucleotides and it gives a stop codon. Exercise number 7. A bidimensional chromatography, pay attention to this please, is performed using a mixture of different human amino acids. This question is yours, Dana. The chromatogram of figure 1 is thus obtained. We have the chromatogram performed using a different human amino acid. This one, the figure 1, is the control sample. So the figure 2 chromatogram show the results of the tetrapeptide hydrolysis of individual X. Figure 3 show the results of hydrolysis of tetrapeptide of another individual Y. So pay attention. Here, they give you the molar masses are shown within the bracket. So for example, this one in the same position is going to be ASP. This is glue, SIR, cis, glide, 3, ALA, tire, val, his, and lyse. All of them, this is the control. So I have to compare all of these, figure 2 and the 3, I have to compare it to the control, which is figure 1. They said, what is the molar of the tetrapeptide found in figure 2? So this one is similar to which one in the control, Yadana? This sequence. Let's put the annotator here. Yes. So this sequence, this one, it looks to similar to what? To which one, the Dana and the control in the position? Dana, can you hear me? Oh, Dana is not here.
Aspirin.
Okay, let's have another one. Let's have Rama. What do you think?
ASP.
Yes, ASP, it's aspartic acid. So the molar mass is going to be 133, right? This is the ASP. The second one, let's move for this. This one, Hassan Watfa. This one is similar to what?
LYS.
LYS. So LYS is going to be what? Lysine.
146.
146. This is the molar mass of this. And then we have to this one, this Hariri.
ALA.
ALA.
89.
So the molar mass is going to be 89. And the last one, yes, yeah, Hadid. This one is similar to which one of the bands found here?
VAL.
Valine. So valine is going to be 117. So they ask you what is the molar mass. What I have to add? I have to add these. I have to add them. Whenever I add them, it's going to be the molar mass. Can one determine the exact sequence of this tetrapeptide? Justify your answer. Can you determine the exact sequence of this tetrapeptide? Can you know, for example, that the first one, this one is, let's say, 146 is going to be LYS. And can you know that which one, this ASP, aspartic? And can you know that this one is going to be 89, this is ALA, and this is going to be the val. Can you know the exact sequence from this figure? Who can answer this question? Can we know the exact sequence? For example, I'm going to start with ASP. This will start with ALA, lysine, valine. Can you know the exact sequence?
Is there a criteria, Oshie?
Yes?
Is there a criteria?
No, you have to know. Do you have an exact sequence? Can you know the exact sequence? Or we have different options?
No. Different options.
You have different options. We have different combinations. Last year, remember probability?
Yeah. Probability and math?
Yes, last year in math.
It.
You didn't take it?
No, we only took the introduction.
Okay, no problem. So probability here for this, we have something called 4 factorial. Do you know factorial? 4 factorial, it's 4 times 3 times 2 times 1. So here we have 4 different amino acids. So 4 different amino acids Maybe the first one is going to be ASP, maybe it's going to be lysine, maybe aniline, maybe valine. So we have different combinations. This is called 4 factorial. 4 factorial, you have to multiply 4 by decreasing order 3, 2, 1. So it's going to be 24 different combinations. So we can determine the exit sequence. How can you explain the chromatogram of individual Y? So we have aspartic acid, we have lysine, and we have alanine. One of these is what? Is deleted, which is valine. So valine is deleted. So it's missed. Okay? So this is about this one. So this is probability. We're going to talk about this in the next chapter about probability. So don't worry about this. So the chromatography show 4 bands. So molar mass, we have to add them. So it's going to be 485, as we did. So they say that do we have the same combination? No, the chromatograph shows us the amino acid sequence. They don't show us the sequence or the exact sequence. So we have different combination, and it shows number 3 bands instead of 4. So one of these bands is missed, and this one is called valine. So here we have maybe the last codon where it changed, and it goes for a stop codon. That's why it's going to be smaller in size. Okay, the last one document is obtained by synthesis of radioactive mRNA and the corresponding DNA strand in the case of certain proteins. So after interpreting the document, show how certain gene mutation might have no effect. So we have here non-coding region. Whenever the mutation occurs in the non-coding that are not buckled or they are not, for example, mixed with the DNA strand. So this is called the non-coding sequence. We call them the entrons. So here diagram shows the DNA buckles don't hybridize with the mRNA. So we have no complementary. So we have mutation in these that do not buckle with the DNA. This mutation, they have no effect. So exons of DNA and entrons, enter and enter. Before export the DNA to the cytoplasm, RNA is subjected to processing. Entrons is going to be cut and eliminated, where exons, they are rejoined together to form the mature mRNA. So any mutation in the entrons will not lead to any effect. So if a mutation takes place in the entrons, it may lead no mutation in the mRNA, in the mature one, since the entrons are eliminated. So such mutation are called silent or neutral. Please join again the live session again. Please re-enter to